Welcome to Flow Stars, candid conversations between Dr. Peter O'Toole and the big hitters of flow cytometry. Brought to you by Beckman Coulter at Bite Size Bio.
Hi, today on Flow Stars, I'm joined by Peter Lopez from New York University, and we discuss the benefit of being semi-retired.
Well, one benefit there is I get to keep my academic titles, so I still have my professor title.
Yep.
And it's kind of a slow easing out of that role while I look at other opportunities.
Pizza.
Although if any restaurants want to hire me, I'd be happy to do a little side job, make a great pizza.
Well, actually, you say that. You actually sent me a picture of your pizza.
Oops, there's one.
And we also take a look at how flow cytometry has helped transform pathology.
Leon Willis had designed flow systems that were mimicking what we had learned to observe under the microscope, but to actually make measurements.
All in this episode of Flow Stars. Hi, I'm Peter O'Toole from University of York, and today on Flow Stars, I'm joined by Peter Lopez from New York University, Grossman School of Medicine. That's a mouthful. Peter, how are you today?
I'm doing well, and how are you, Pete?
Oh, no, I'm really good. Thank you so much for joining me today. I've known you for many years, but it's really great to actually probably to get you to know you better than I actually know you, if that makes sense. Because when we were at work conferences, meetings, the conversations generally go personal to lots of work, whereas actually here, probably get to know a bit more about you than I realized. And actually, the first thing before we started, you mentioned you're semi-retired.
Yeah, this happened. People say that to me with kind of the same inflection in their voice, with a little bit of a question mark at the end. I've been at this for 46 years. And it's been, I've been thinking about this. The pandemic is one thing that I think factors into a lot of people's, career or life decisions now. I thought that this was a good time. I am officially in the U.S. Retirement age. So I did a kind of a staged retirement. So at the moment, I'm semi-retired. That happened March 1st of 2023. And I stepped down from the director's role as the flow cytometry laboratory director. And I'm staying on board for a day per week. I kind of wanted to do that to keep, one benefit there is I get to keep my academic titles, so I still have my professor title.
Yeah.
And it's kind of a slow easing out of that role while I look at other opportunities, because I didn't want to just cut it short and say I'm done. It's been great, and I'm out the pasture.
I've got to say, this is for those listening, go and flick to YouTube briefly, because Peter looks super young, and that is why it's so nuts. But then, just stupidly, I did look at some background, and you studied your first flow cytometry is 1977.
Yeah.
Correct.
Yeah.
So that must have been the birth of flow cytometry at 77.
Well, things were already happening, and to be straightforward here, my first job was in a lab at University of Rochester in Rochester, New York, where there was flow cytometry instrument development going on, and that was something that. Yeah, it was relatively new. The stuff that was going on. The first patents in flow cytometry. It depends on if you consider a Coulter counter a flow cytometer, and I do, because it's a cell measuring device and it flows cells. And that patent was, I think, around the time that. About when I was born, it was when that patent came out by Wallace Coulter. Or if you want to go to, kind of the more straightforward fluorescence-based systems. That was Wolfgang Goethe's patent when he was at FIVE before Partech, and that was in the 1969. So yeah, it was kind of early on, but you were hard pressed to find laboratories that had cell sorters at that point. Although at the University of Rochester we had them, we had one, but that was not the group that I was working with. I was working with Leon Willis. And it was, I mean, they were doing stuff that just got me so excited, with my background and the things that I had interests in. I was a young man, and I was quite lucky because that job was almost a recruitment. So it was my first job, and I didn't have a job in the field, and yet Leon Willis came over to me and asked me if I wanted to work in his group. And I was like, hey, that was kind of a dream for me. As they said, the rest is history.
So what was your education background before 77? And what did you just come out of before you took this role? So this must be your first job.
Right. So
professional job, sort of career job. Maybe that's a better way of wording it.
Right. Yeah, because my first job was cooking in a restaurant, which I still do. Not in a restaurant, although if any restaurants want to hire me, I'd be happy to do a little side job. Make a great pizza.
Actually, you say that. You actually sent me a picture of your
pizza. Oops, there's one. Yeah,
that looks like I'm just a big giant pizza head.
I might use that for my background for now on for my Zoom calls. I like the pizza head, Pete. It
works quite well. Yeah, but pepperoni head just makes me sound like probably my complexion sometimes. So go on. So
so my background was really just basic biology background. I got into a program. Frankly, I really had no idea what I was getting into to learn cytopathology in a clinical setting. I honestly didn't know what I was getting into, but I thought it sounded cool. It involved microscopy, and I already had interests in optics in the other direction, telescopes. So looking at distant things rather than looking at small things, and I was born in New York City, and New Yorkers in New York City have a very different worldview. I mean, everything for a New Yorker is New York, and outside of New York is who knows. I basically went to Upstate Medical Center in Syracuse. I had no idea where I was going. Most people were showing up to the university in a car with family, so forth. I flew in with a backpack and a guitar because I thought that's what you're supposed to have at college. I mean, I did have other things coming eventually. Stuff got moved from my bedroom to my dorm room. But I got involved in this program, which is basically studying alongside the medical students things like pathology, things like medical technology, things of that nature, with the goal of learning how to do cytopathology with a license. So I basically came out of this program with a clinical license where it was all microscopy, 100% microscopy. I mean, we were with our eyes glued to the microscope probably 5 hours a day, learning how to look at essentially pap smears and to do a pre-screening of these things in order to screen out potentially malignant cells. Go ahead, Pete.
Yeah, so I'm intrigued now. So you got into microscopy, which is where I started, and then I got into cytometry, but I've kept microscopy and cytometry. But as far as I'm aware, you kind of dropped a lot of the microscopy and went major in cytometry, or did you keep some of your microscopy going alongside the cytometers?
Well, that's kind of where that first job came up because, Leon Willis had designed flow systems that were mimicking what we had learned to observe under the microscope, but to actually make measurements. So, classic cytopathology in the early days was really kind of a subjective, you looked at a lot of different cell types, you learned how to describe these things in detail, what you were seeing, the nuclear conformation, the chromatin, the size of these cells, staining patterns and so forth. But in the 50s and the 60s, Papanicolaou came up with this early work, but one of the pioneers was a man named Leopold Koss, Koss. He is actually in New York initially. He was at Montefiore Hospital, which is part of Albert Einstein, in the Bronx. And he wrote the book on diagnostic cytopathology. But there wasn't this kind of metry part. There wasn't this type of measurement really being, written down and studied. It was more of, look at the patterns, look at these things, and you could very reliably make these diagnoses. At Rochester, there was a guy named Stanley Patton, also a famous cytopathologist, and he was the person I studied under. What he wanted to do was to try to actually do cytometry, to actually make measurements and to see if certain cell types have measurable characteristics. And this was just done visually. This was not done with any kind of DNA staining, any kind of special stuff. And he, it's pretty cool because he was, this was in the 60s, and he was making cellular measurements. You could make as you very well know, you can very easily make a measurement on a microscope with a stage micrometer and a calibrated eyepiece. He went a step further and started weighing stuff. So he would trace images of cells and nuclei. Yeah. And he would start making weight. He was making weight determinations by cutting these things out and putting them on scales and stuff. But he gathered enough information that served as the basis of this project. So I kind of went off a little bit on the story. Keeping the microscopy, I think, really played a very big role for me in flow cytometry because I got into that part of the field, knowing a bit about what we were actually looking at as the cells, as things were going through the machine. So I still will say this, to any young flow cytometrist or people already in the field, that a microscope is your best friend in a flow cytometry laboratory, and you really should have one. And you should really become very comfortable with turning that thing on and using it, because it's going to get you out of many binds where just looking at dots on a dot plot, you can say a lot, but sometimes the picture's worth 1,000 words. So I kept with it, and in fact I did actually when I was at the Aaron Diamond AIDS Research Center, and actually even when I was at Harvard, I did have small microscopy laboratories that I was simultaneously overseeing. I mean, we had a very early one of these kludged together deconvolution systems when I was at Rochester, before they became, people started making them prettier and more reliable. So I had one of those when I was at Harvard at Dana-Farber, and then later on at Aaron Diamond AIDS Research Center with David Ho, we used one of these. I don't know if the name's changed, but it's called a Delta Vision system. And it's a deconvolution microscope.
Yeah, you still get Delta Vision. Oh, sort of. Still get Delta Visions. Okay. And the OMX, but they're things are moving forward still, as always, with the pace.
Yeah. So I kind of like that aspect of it, and I think I really encourage people to really become very comfortable with the microscope in a flow cytometry kind of a setting. I could talk for quite a bit on this, but as can you, I'm sure. Where you're looking at something on the screen and you know something's wrong. And you're trying to describe it from what you're looking at on the flow cytometer. And my first thought is, let's take seven microliters, which is, by the way, the amount that fits really nicely under a 22 millimeter cover slip. If you take seven, that one I found by trial and error. It's almost like every time you make that slide, a wet prep, it's gonna, you're not gonna end up with a lot of bubbles and stuff.
Top tip number one, we need that for the short edits. For the 7 microliters being the perfect amount. I have you mentioned during that point, David Ho. And so I'm trying to, one of my questions was, who's inspired you? And you sent me a picture of David. So I presume David is one of your people here. So I thought showing the pictures actually to give some credit to David as well.
Just a brilliant, wonderful, I mean, the depth of his knowledge, David Ho was the person who was the president of and actually the Aaron Diamond AIDS Research Center. Aaron Diamond is a real estate developer in New York City, and his wife is very interesting also, Irene Diamond. They did a Vanity Fair issue with Irene Diamond as on the cover, and I think the title was "Rebel with a Purse," because she had the money from his to go after really socially important things. She was a playwright back in the early days, the early Hollywood days, and their interest in the HIV situation came because she was noticing, some of these artists were dying from this disease. So David was brought in at the start when they formed the Aaron Diamond AIDS Research Center, and he's a very understated individual, but his reach is worldwide. I mean, he has, his group came up with the antiretroviral cocktail that turned HIV/AIDS into, from a death sentence into a chronic manageable situation. I was lucky to be invited to his 70th birthday, which was also the 30th anniversary of the Aaron Diamond AIDS Research Center. It's a fantastic. I mean, I've never been to a gala like this. I mean, this would have, if I had a tuxedo, this is what would have been the place to wear it, but I don't have one.
Is this you at the same do? This isn't you at the same do though, is it?
Well, I'm sorry, say again.
This isn't that party though, is it?
Oh no. This is
about to you both looking super informal in a jacket. And
No, this is one of the. I had already moved on. So that's another really nice thing. I quickly felt like I was part of this part of the family. With the group, the Aaron Diamond AIDS Research Center is a small center. I don't think there are more than 100 people working there. So the interactions were fantastic. I mean, thanks for noting that. I mean, David's been, so helpful to me. I mean, I've been lucky. I've had a lot of really great mentors. I mean, I could also, mention, Leon Wheelis, who got me started in the field. After working at Rochester, I moved to Philadelphia and worked with Donald Mosier, who's a world-class immunologist. He really let me. He gave me a lot of freedom, and that was the first commercial flow cytometer I ever used. So I was moving from home-built instruments that we made in Rochester. And in fact, just, maybe it may also be of interest, we had developed what I believe was the first imaging flow cytometer in Rochester. That machine was built already when I started in 1977.
That wasn't this, was it?
No. This is actually two Coulter Epix cell sorters that were, this was the cell sorting complement. We had two in that laboratory, although there were other laboratories at Dana-Farber. I think a lot of people know of John Daly. John Daly also had another core laboratory at Dana-Farber. We had we weren't competing. We were just serving different groups.
How is John? You still in touch with John?
John's retired. I think he's actually the same age as I am, but he took his retirement, I think, last year. And from what I have heard, he's building small. Well, I don't know small, but he's boat building. He's off building boats somewhere.
I remember looking at some of his Facebook when he retired. So on Facebook, it's the only way you keep in touch with some people is via Facebook, and playing the fiddle, I think, over in Dublin at one point and going around. It was I just wondered how it was. So looking at this, and another instrument here is
that's the FACS-2 instrument, which is very similar to the instrument. This was the first commercial flow cytometer that I ever operated. This one, I think this is the system that's, I believe, at the Smithsonian. So it's in a museum someplace. I'm sure Paul Robinson can tell us more about where this system came from. But that's it. I mean, that it's a monster, and that thing was only capable of doing two colors. And the only way you could do two colors was with two lasers. All of the dyes that we rummage through when we're trying to figure out how to put together 30 color experiments. This stuff was unheard of and unimagined back then. I mean, when phycoerythrin came about, people were thrilled. People were. That's actually a funny story because I was working with Don Mosier at the time. I worked with him from 1982 to 1986, and he was a world-class immunologist, so he had connections with Herzenbergs and Becton Dickinson and so forth. And Don handed me a sample of phycoerythrin with the spectra, and he said, BD, or I don't know if it was BD or Herzenberg or whoever, they sent the sample to him, and I don't remember what it was conjugated to or whatever. He said, let's give this a try on the machine and see how it looks. So I had no knowledge of this. This is before, all I knew is this is a new dye. Here's the spectra. And we had a two laser instrument, and two laser instruments were rare at the time. So my thought was, they're sending this to us because we have a krypton laser, and a krypton laser puts out a really strong 561 or 568. I forget the nanometer line. And I said, okay, great. I'll turn this on. I got a filter, an optical filter, so I could look at it. And I ran it, and I told Don, I said, this stuff is screaming bright. Look at this stuff. It makes Fitzy look like you didn't turn the lights on. And Don said, fantastic. I'll send the information back. So they sent the information back, and then sometime later, Don comes in and said, Peter, you didn't do it the right way. You didn't run the sample the right way. I was like, what do you mean? And I said, they didn't want you to run it at 561. They wanted you to run it at 488. And I was like, why would you want to run this at 488? And, like, now you can get another color off the 488 laser. So you don't need that other laser. And again, there's another one that the rest is history.
John, what's amazing? I mean, that if for the listeners now, you're thinking about how exciting it must have been to see PE for the first time. We just take for granted now, but to be at that early innovation. And yet there are still those moments in the lab, even today. There's new dyes, new technologies, new approaches. And I don't know, I think the last 3 years, there's been two moments that I've been looking at whole new beta test or concept products and just gone, 3 times. And just gone, oh my goodness. Who'd have thought we could ever do that? And it's just different things that just were not possible, not imaginable. Then you think, wow, what science can we answer using it? So imagine now going from one color, two color to have more colors. It just opens up what you can do. And now they're asking for 30 plus colors.
No,
Guys,
sure.
just no pleasing some of the scientists today. I have a question though.
Yeah.
I've been listening. You. How many different jobs have you actually had?
Oh, well, yeah, I've had a couple, but you got to remember it's over 46 years, so it's not. Yeah, so uh, five. I started at University of Rochester in '77. I was there from '77 to '82. Then I worked with Don Mosier at the Fox Chase Cancer Center in Philadelphia, '82 to '86. Then I was by the way, I was recruited. This is another odd thing. And I think, well, I'm gonna get back to this, but this has a bit to do with Paul Horan, because Paul Horan is another guy that I worked with in Rochester, responsible for the PKH dyes, the cell type. That's his initials, Paul Carl Horan. He's been also another one. Although I never worked directly with him, we were all in the same group. But I think he used to drop my name at meetings or something, and I'd get these phone calls from people, and they wanted. I was like, "Hello, who are you?" Because I didn't know. Well, actually, a lot of the times I did know the folks. But getting back to the jobs, Fox Chase Cancer Center in Philadelphia. Then I went to Harvard, so I was at the Dana-Farber Cancer Institute. That's where John Daly was working in the other lab. But I was working with Ellis Reinhertz, who is big T cell immunologist, responsible for a lot of the T cell receptor knowledge that we have. So I was there for a while, and then at that point, so that was, let's see, I'm not great with math. I used to be. I'm just trying to think of how long I had been in the field. At that point I had been in the academic environment for quite a while, and I heard, there's another world out there, this industry. So I said, well, maybe I should check out industry for a while, see if that like freshens things up. That was there was a company at the time called Cytometry, and they were doing demos for at that point they didn't have the MoFlow. They came up with they commercialized the MoFlow cell sorter, but they had other instrumentation at the time, and they were demonstrating stuff. And I was actually, John Daly used to get the demos, and John would invite me over, so I'd go over and look at the stuff. And after a while, they said, "Hey, would you consider moving to Colorado?" So I did, and I worked on the MoFlow for a while. That went until 2000, so that was '96 to 2000. And then at that point, moved back to New York. I'm a native New Yorker. And worked with David Ho from 2000 to 2007. For about the last 2 years of my time with David Ho, NYU was aggressively trying to recruit me. And that was an interesting story because I was playing squash with. I'm not a great squash player, but I was playing squash with a lot of the senior faculty who I grew to know from previous jobs. A guy named Mike Dustin, who is another famous immunologist and character, and you probably know him because he's at Oxford now. I knew Mike when he was a graduate student. I worked with him at Dana-Farber when he was a graduate student. I mean, talk about another great mentor and just prolific scientist. We'd be playing squash all the time. Dan Littman, also another big immunologist, and I'm playing squash with these guys. And meanwhile, they're trying to convince me without, really just not high pressure to come across the street. And that's what it was. It's basically across the street in New York City. At the time, Aaron Diamond was on 24th Street at First Avenue, and NYU is at 30th Street on First Avenue. So it was basically, a job. It wasn't improving my commute in any way because I live 60 miles north of Manhattan. Well, maybe it did by 4 blocks.
Peter, out of all these jobs, what have you found the most difficult time in your career to date? What was the hardest time, the most challenging time?
Wow. In a lot of these jobs, the beginning of the jobs were the challenging times because at Fox Chase, it was very challenging because I had never operated one of these commercial instruments before. So I was a little challenged at the beginning, getting things up and running reliably. I was really learning on my own. I don't know if it was common back in those days to have service contracts or to have people helping you, but I know we didn't have one at Fox so I had to figure out the facts too on my own. I did reach out from time to time to folks at NIH. There was a person there named Susan Sharrow. Susan was another high-level immunologist, and she had a number of systems, and she had a great relationship with Don Mosier, so I was able to chat with her and to get help. So, and then both Dana-Farber as well as NYU, I think it's safe to say those core laboratories were in pretty bad shape when I got recruited. So it was tough going during those early days. So I think that would be, a couple of these jobs that I took in the early stages were rough because I didn't have a lot of resources. I just had to, figure it out, make things happen. I mean, NYU just really was difficult because the lab was a disaster. At the time, it was a single-person laboratory. Right now, it's a 6-person laboratory at the same institution. And that's another story because a lot of the work was a lot of the NYU work was coming to me at Aaron Diamond because they just couldn't get work done.
So Peter, why keep jumping? Because every time you jump into you get a place running well, smoothly, you're on top of the instrument, and then you've hopped and you've gone into a place that needs pulling up. What's motivated you to keep switching?
Well, maybe there's just a little bit of ADHD that I deal with in my life. So I'm always looking for something shiny to distract me. It may really be that, after, it sounds like a lot, but these jobs were 7 years, 10 years. I mean, this NYU job, which I'm now retiring from, I've been there for 16 years. Just new challenges. When I was at Dana-Farber towards the end, I started, saying, well, yep, been there, done that. What's my next move? And sometimes these things happen, faster than other times.
Okay.
And that was really it. Just and these were moves in location also. So that was another thing that you find a place to live, and maybe after some, maybe the neighborhoods start changing or things start changing where you're living, and so it was. I think these were the things I was never. As I said, these were all recruitments. So, I mean, maybe that's another part, when something gets, gets dropped off at your door, you look at it and say, maybe this is something I want to keep.
An element of, it's not flattery, but feeling wanted and
could be
going where that's wanted. So I'm going to ask you the converse. I asked you most difficult, most challenging time, and that was the start of starting up the labs again and the starting of a new lab, bringing it up. Of all the time in your career, what has been if you could go back and relive a year, which period would it be?
Wow. When
you've got glasses on, you can look at those rose tinted glasses and think, they were golden times. What was the most golden time?
Well, Cytometry in Fort Collins in Colorado was, it certainly had its ups and downs, but there, because it was a smaller group, and I may even segue into Aaron Diamond also, because they were both smaller groups where I had a feel, definitely at Cytometry. I mean, once things started clicking with the MoFlow, I was feeling like we were just kicking ass and just making real important contributions. I mean, we certainly, with any new flow, any new instrument, no matter what it is, there's going to be some issues, but we were addressing things, we were getting stuff resolved. People were happy with what we were doing, and I felt, this was, there were really late nights at Cytomation. Yeah, I'm talking about work. I'm not talking about the other.
Yeah.
There were other late nights too, which were always fun too. The camaraderie of the group really just turned me on. I mean, everybody is willing to stay till midnight. It's not just me working in the lab, with a light on and nobody else around, but people were just, the camaraderie and the notion that we were doing significant stuff. That was really important. And I have similar kinds of feelings. All of my jobs, I think there were these times, but to answer your question, the standout moment I think would probably be those days at Cytomation.
I love the fact of all the cytometer pictures you sent me, you didn't send me one of a MoFlow.
I didn't send you one. Okay.
No, I'm just looking through. I can't see one of a MoFlow, and I can't find mine.
Okay, don't worry about it. Don't worry about it.
Change tack a bit. You said you burnt into the late nights, and you sent some pictures to describe yourself. So after a late night, what you sent me this picture of these two drinks in kind of really cool sort of Art Deco glassware. Actually, I've got to say, I love the glasses. What are they? And are you that famous for drinking?
Well, so the glasses actually, that's a bit of a story. We've been to meetings together, and people know me. I'll enjoy an adult beverage with colleagues, chatting over anything at any time. One of a colleague and now a friend at NYU, actually someone who invited me to help them on with a really significant cell reports paper. He after he left. He's a consultant and working for and doing other things kind of in bio and also in business. He got seriously into cocktails, and he started sending me pictures of cocktails, and he'd send me a picture of a glass with something clear in the glass, and I was like, oh, that looks like a martini. And he said, no, it's a daiquiri, and I've cleared out everything. And I was like, and I said, yeah, and how's it taste? He says it tastes exactly like it should. And so this guy was in very deep. And we got into it at my house because he would send me recipes. He would always tweak a recipe. So with this, what we're looking at here, first off, the glasses are a gift from my wife, Linda. Surprisingly, this is a Christmas gift, and that year we both gave each other cocktail glasses. These are the ones that she got for me, and yeah, they're really very, very special. Hand washed, by the way. And
obviously, yes,
yeah. And what you're looking at, and this is simply a picture that I took to send to my colleague Vish, and this is a what's called a maple sour. It turns out that I've got a lot of hobbies, and one of them is that I make maple syrup. And
there you go.
There it is. That's my 2023 crop, which is the season is over at the moment. But the last so that's the result of a number of different days of collection of sap and boiling sap. Yeah. Oh, and that's my house. And actually, let's see if you you can sort of see in the tree to the left at the base, there's a little bucket. And actually, if once you identify that, you can see there are other buckets in the pictures. They're little white buckets at the base of the trees. Yep. There you go. There you go. Yep. And that's for collecting the trees. There's a lot of trees on this property, but the selected trees are sugar maples. So the sugar maple sap is then boiled down. And this picture, I think, is maybe no more than 2 weeks old. So So I've got a lot of maple syrup. I love maple syrup. I mean, that's just pure pure maple syrup is a joy. I mean, that's my friends in Canada call it liquid gold, because Canada is such a big producer of it. Yeah, so that's a maple sour. It's you know what? I'm not going to be able to. I know it's got maple syrup, and I know it's got lemon juice, and I think the spirit is thinking it's vodka, but I'm not sure. Or it might be rum. But anyway, that's what it is. And these are just pictures that go back and forth between Vish and myself. He's always got some really amazing stuff that he's doing, pulling together, and I'm just sending him kind of this everyday stuff. But I try to get nice glasses.
But at least the pictures are sensible that you're sending between the both of you. Peter, I'm going to ask you some quick fire questions.
Oh boy.
Okay. So, what's your favorite color?
Oh, I may lean towards red.
Okay. What's your favorite dye?
Oh, my favorite dye. I always wanted to say rhodamine in a public setting, but it isn't, because I like that rolling R, rhodamine, but it's not. I like the phycobilly proteins. I'm going to go with PE. I just like big molecules that fluoresce like holy hell.
Okay. Are you an early bird or night owl?
Early bird. You said am I an early bird, right? Yeah.
Are you an early bird or a night owl?
Oh, night owl. I'll be burning the candle late. So
night owl.
Yeah.
Okay. PC or Mac?
PC.
Okay.
Yeah.
McDonald's or Burger King?
Neither.
So if you had a takeaway, what would you go for?
I'd try to find some place that has a good selection, but but I can identify what's inside.
If you were to cook yourself, what would you cook?
Oh, pizza is usually one of my go-tos. I will be eating pizza at my house, like the one that the image, at least once a week.
Okay, yeah, we're definitely a pizza house once a week. Yes, definitely, for sure. Coffee or tea? Tea.
Coffee, but this is tea.
I was about to say that's no way is that coffee. Anyway,
this is tea.
It's coffee in the world. So okay, so you got tea now, but not generally coffee. Beer or wine?
Oh boy, that's a tough one. I'm gonna probably say wine. That's
okay, that's fine. That's fine.
Yeah.
Red or white? Red or white wine?
Probably gonna be red.
Red. Okay, I'm now going to challenge you even more. Wine or cocktails?
Wine probably.
Ooh. Okay. Chocolate or cheese?
Oh boy, together. That's a probably cheese.
Okay. Well decided. So you're married, you've got Linda, your wife, you said. Who cooks at home mostly?
I was trained professionally. So I'm my wife can cook, but it's me.
Okay. TV or book?
Oh yeah. It's probably could be TV.
Okay, and God, what's the trashiest TV you dare admit to? Or you daren't admit to, but you're about to tell me anyway?
Trashiest TV. Oh boy.
Come on, you can't be watching that much bad TV to not to be able to decide on one.
Trashy TV.
The smile on your face tells me there's.
I'm kind of liking, and she's from Britain. Kunk.
Kunk on Britain and stuff. Yeah, okay. Okay.
Would that be considered trashy? I think she's.
I guess you could get away with trashy. Very entertaining.
Oh man, are you kidding me? I mean, I have had the longest laughs with some of the. I mean, her timing and some of this stuff is just absurd that she says. but sometimes she comes up with something and I just can't, I have to turn it off. It's like, nah,
Do you know what? We should do a Kunk in Britain for a flow cytometrist or something. You just ask those really daft questions and just take it in a direction not meant.
Right. So science, man.
So, oh, I love it. What's your favorite film?
2001: A Space Odyssey.
Oh,
If there's anything better, I want to know about it.
good classic film. Are you a Star? What would you prefer to watch, Star Wars or Star Trek?
Oh, Trek. Star Trek.
Good man. It's kind of a trick question, but some people do actually admit to Star Wars, but surely scientists got to be Trek.
Cool.
What sorts of music genre are you into? What's your music taste?
Music tastes are usually I'm a child of the late 60s, so rock and roll. But I will guilty pleasure, I will enjoy pop, kind of pop rock type songs. Yeah, that's almost everything that I listen to. Although I have a classical music collection. So if you're going to ask vinyl, CD, or streaming, I have a lot of vinyl. Although I don't have my vinyl set up, but I'm a bit of a I like that warm sound.
Oh no, thinking of vinyl records, I don't know if you know Scott Fraser. So he's a very microscopy world. So I do the microscopy, so similar to this.
Yes.
Scott makes his own amplifiers, and he's got his vinyl record player is made on an IKEA breadboard.
No kidding.
And interestingly, I was with Scott there, actually, I was going to mention earlier, we talked about all the technologies that have moved through. So Scott brought spectral microscopy to the scene. So spectral are mixing the 32 detector array, actually with Scott Fraser's baby that he brought into the confocal microscopy world over 20 years ago. And here we are 20 years later, only just really embracing it in the flow cytometry world.
I know.
What do you think to spectral flow cytometry?
Well, I think well, first off, I mean, you already prefaced this by saying it's been around, and it's shown, on the microscopy side, and some people will even point to the astronomical work that is done with spectra, which is another kind of that was another hobby of mine a while back, but I think there is great power there to do things. People might say, well, yeah, you could probably do that without the spectral technology, but it's empowering having that capability. Things related, definitely anything related to autofluorescence. I think that's it's a strong help there. I think it's there are some things that I'm still waiting for people to show me if how for instance how I don't know if this is out there already, but how does spectral work with dyes that shift their emission, and then I may just not know the answer because it's out there already, but it's not obvious to me how you could do an experiment like with Indo 1.
Yeah, I say Indo dyes would be
yeah.
And do you know what? Conceptually, it is possible. But it would take offline.
Okay, thank you. Like I said,
exactly that can work.
Right. Yeah, I'm sure it is, and I'm sure at some point, even if it's not in place in flow cytometry, if enough people ask for it, that'll come out. But I think it's I know some labs where they're just replacing everything with spectral systems. I don't think that's a bad move. But I do also think that there is a place for conventional systems, especially if they're open and you can get into them and you can do things with them. That was a real a real strength of the MoFlow. You know, a lot of people would look at the MoFlow and they're like, oh, look at this thing. It's a whole bunch of, it looks like somebody's physics project. And we knew that, but we never put a box around it. It was just, you know, people want to get in there and do things. So I think there's still going to be a place for that. And maybe that's where I'm not going to put a crystal ball up and try to make predictions. But, you know, maybe we might see instruments, you know, I have no knowledge of this, but, you know, I'm just putting it out there. Maybe we're going to see flow cytometers, you know, there might be, a flow cytometer that's just an open machine again, you know, where you know, where most of your instruments are spectral, but here's this other one if you want to do something special, and you can actually, you know, change the filters, or you can, put something, you know, you can put a half-wave retarder in front of the laser. You can do something that, you know, would be impossible with these nice clean boxes.
Well, strangely, that's the way the world, the microscopy world is certainly going down the homebrew type microscopes as well, sort of the homebrew specialist things, which would be to answer just that sort of question. And I'm thinking of homebrew, I don't mean alcohol now.
Which I do. Oh, and that. Oh, there you go.
You also sent me a picture of
it's a telescope. Yeah.
A telescope for stargazing and stuff. Yeah. And so this is what got you interested in optics to start with. Is that correct?
Yeah, it is. I was quite an astronomy buff. I mean, to be, you know, to bring it back to the beginning, my father, who actually never graduated high school, he was in the HVAC, heating, ventilation, and air conditioning, initially as a mechanic, and then later as an engineer. He when I was a baby, things were happening with space flight. You know, the Mercury astronauts and so forth. And my dad used to play games with me about where we were making believe we're launching a rocket. I mean, he will tell me these stories because I don't. He's passed on, because I don't remember them. But I was some early at 3 or 4 years old, and talking about putting liquid oxygen into the lower stage of the thing, and we have to wait till it vents, and you know, I don't know how this happened, but I it's really quite the science art. And then that space thing got me interested in astronomy. I grew up in New York City. And there's a famous planetarium called the Hayden Planetarium. It's attached to the Museum of Natural History. And I wanted to go to that, but I was too young. They wouldn't let children in unless they were 5 years or older. And I must have been 4, because I kept on saying, I want to go to the Hayden, because you could go to the museum. The museum had, you know, dinosaurs and all this, you know, all this other cool stuff, which I was into too. But I wanted to go to the planetarium, and finally I turned 5, and my father said, we're going to the planetarium. And that was just so thrilling to me. So I was interested, that got me interested in astronomy. I was in astronomy clubs and stuff like that. And then, an outgrowth of that is having your own telescope. So, I read about it, I learned about it, and the optics of putting that together got me that wasn't my telescope. That's a very similar one. Mine is, I still have it, actually.
I was about to ask, do you still have your telescope then?
I still have it. And I really have to get off my ass and pull that thing out, because I've been carrying it around with me now for a while. And if you remember the picture with that big white tube, it's not attached to the right. Yeah. So the big white tube is the telescope, and everything underneath is what is called the mount. It holds it. So they're disconnected, and the tube in my garage is vertical, and people look at that thing and think it's a water heater because it's so big and round. It looks like, a domestic water heater. I was like, no, it's a telescope. So I've been saying that long enough that I really should get that thing out and look at it. I'm north of New York City now, so the skies are pretty nice. And I'll get out there still with binoculars and see, try to see things. I'm actually a solar eclipse chaser also. I've seen a number of total. I will travel to. I'll get on a plane if it's a reasonable trip to go see a total solar eclipse, and I really encourage people to make the effort if in their lifetime just to see such a thing. I mean, a total eclipse is just the most bizarre spectacle.
That is a total fluorescence geek thing to do, to try and remove as much scattered light as possible, and you just chase the removal of scattered light. I noticed, actually, if this is your house, you've got a lot of trees around it, which is going to give you a problem. Now, we've only got 5 minutes left, and there's a few things that came out, some of more of the pictures. You sent me a pictures. So these mushrooms.
Hobbies, yeah, that's a hobby. That's these are oyster mushrooms that I've grown. They're actually a little bit past prime. They're starting the outer margins are starting to curve up a little bit. I started doing this. This was my pandemic hobby. My wife Linda is a cancer survivor, and she's been interested in increasing mushrooms in our diet, and been buying them at the store. And then I learned that you can actually grow these things on coffee grounds. Again, coffee first, tea later. But the thing is, they're quite easy to grow. They grow incredibly fast. It's quite frightening how fast they grow once they get. So I don't grow them on coffee grounds. They grow on straw, and that's on a scale. I think that's a pound, almost a pound and a half of oyster mushrooms, and they're delicious. I mean, they're just fantastic. Yeah.
My nephew, actually, this is his living. He's growing mushrooms for fine dining. Sells them down on the London markets. So Harvey Piper down there. So he's got things like lion's mane.
Lion's mane. Yep, I've grown those. Yeah, I've grown lion's mane. I don't have a good picture of the lion's mane. Yeah, I grow lion's mane and these currently, but I might try to broaden up a little bit.
So you've got cocktail making, maple syrup. You've got mushrooms. You sent me a picture of this.
Oh,
spider.
That's it's interesting because it's a Fiat spider. The Fiat 124 spider. Actually, to be really precise, Fiat stopped selling their cars in the U.S. in I think it was 1982, and then another Italian company called Pininfarina basically took the same exact car, slapped their name on it, and sold it for another 3 years in the United States. And as of '85, they went away. Actually, they've come back. But anyway, this is a 1983 Fiat Spider. It's a restoration project that I do drive, but it harkens back to my first car. I had the same car. I think it was a 1969. And that car was a death trap. It was completely rusted. And after I got rid of, actually, that just went to the dumpster. I mean, it just got picked up by a flatbed, and the first one. And then, years later, I said, maybe I should find one that still has some metal and it's not completely rusted, and try to keep it up. So that's my little baby. Yep.
So, do you have old cars? You've got your syrup, you've got your mushrooms, you've got your telescopes. You confess to working late at night. You've traveled many different destinations, and you have a wife.
Mm-hmm.
How does that work? Where's the time here?
For those of you who we see at meetings, you usually notice that she's there with me. So almost all of my traveling, I will include her in it. I'm happy to say that, that's one thing that I really learned through the pandemic is if there's anybody that I would wish to be locked up with for years on end, it's Linda. So she's just, she's, as they say, clearly the better half. She's wonderful. I can't say enough about her. And she puts up with all of it.
The better half, but not the better cook. Can I quote you on that from earlier?
Yeah, probably. It's okay. She'll be fine with that. She'll be fine with that. Yeah.
So we are up to the hour, but I have to ask one more question because you sent me a picture of. Please describe this. This looks like a torpedo. No, it's a submarine. What is it?
Yeah. So this is what's called an autonomous underwater vehicle. For those of you who were at the Cyto 2022 meeting, there's a woman kneeling on the right side of the picture. That's Heidi Sosek. She was one of the speakers. Not in the, I think that's your mouse. Not in the solid blue. To the other side. Yep. That's Heidi. Yep. That's Heidi. So this is at the Monterey Bay Aquarium Research Institute. I collaborated. In fact, I dabbled a little bit with marine biology, this is an instrument that they built and designed, and you basically put any type of scientific payload in the orange part in the front, and send these things off, and they run missions autonomously. They're robots. And they can come up for energy recharging with solar panels. So this is a group. These are a number of people who were invited for a workshop, and we were basically challenged by the Monterey Bay Aquarium to come up with apparatus to stick into these things to do things like predict harmful algal blooms and things of that nature. So my job there, Heidi's a lot more. I mean, Heidi's one of the biggest names in this field. I was just there because I got a little bit more on the instrumentation design side. We were the flow cytometry folks, but there are people there with all sorts of novel. I mean, there were microscopists in that group, looking at how can we get images of things that might predict. So that's that was a lot of fun also, to go back to your question about highlights, but that was just a side kind of a gig. But
Peter, we are over the hour, and
okay.
We didn't get to talk about Isaac and the importance of Isaac and the recognition to yourself that Isaac's awarded you some of the courses and events and some. Well, you've got to work out which one you are on this picture because
it's the guy with the longest hair.
Yeah, you had super long hair at one point, and I think you had
yeah. So Leon's in the Leon Willis, the guy who gave me my first job, is seated next to Ted Young. Leon's in front of Myron Malamed, but if we're not if those names aren't familiar, he's the guy in the left. There's both guys on the left have kind of tan colors. So Leon's Leon's the guy with the facial hair.
I just love the fact just looking at how you changed over time is utterly brilliant. So I couldn't resist actually putting those up. Peter, we are really sorry out of time. I've still got questions to ask you, but you at Cyto this year?
I will probably be at Cyto this year. Yes, I will.
We shall grab a drink.
Yes, absolutely. Yeah.
And some mushrooms. Because you're a fun guy to be with. It's a really bad joke to end on. Anyway, so everyone who's watched and listened today, thank you so much. Please go back and look at the other previous Flow Stars where some more history is there. Yeah. Hopefully see you all at Cyto. And please, Peter, yourself, thank you so much for joining me today. You've been brilliant. Thank you.
Thank you.